Hello and welcome to mitosis. Mitosis is something you've heard about before, I'm sure, so we'll swing by this, but there are some phases I do want you to know. So mitosis, has a lot of different roles. You could be thinking about a child that's developing, and here's a picture here where you would have these cells that are duplicating. Or you could have a scrape on the knee where these cells have been damaged and they need to duplicate and you need to create more cells. Or even cooler is organisms that can generate a whole new limb. Now that'd be really cool if we could do that also. So those roles are for growth and replacing dead cells. I wanted to show you that the bacteria and the archaea actually don't fall into the same pattern of mitosis. So let's look at that first. They don't have a nucleus, right? Bacteria and archaea don't have a nucleus. So instead, they have their DNA in one big coil like this, in one big loop. So what they have to do is reproduce that coil, make two of them, and then slip one over to one side of the cell, slip another one over to the other side of the cell, and create two cells. This is not really called mitosis. It's called binary fission. So binary fission, you think of the archaea and the bacteria. Okay? They, side by side, you have the archaea here, the prokaryote cell with a big loop of DNA, and your cells, which are eukaryotic cells that have a nucleus, and the DNA would be inside that nucleus. So first you have to dissolve the nucleus, release the code, copy it. It's in steps. I'm going to show you what happens. When you look at eukaryotic cells, they could look like this. Very different, right? You have one here that has a nucleus. It looks pretty normal. This one has all this squiggly stuff instead of a nucleus. And what you're seeing here are the condensed chromosomes before cell division happens. The generic term for that is chromatin. So chromatin is all the genetic material that you have in a cell. It's not visible when the cell's not dividing, like in this cell 1. It is visible. It becomes visible when the cell starts to divide, like in cell 2. So clearly they look really different. You could look at under the microscope and see all these different cells that look completely different. One that has a visible nucleus, another with a nucleus, another where these chromosomes seems bunched up, no nucleus. Here these chromosomes are moving away from each other. So clearly they're in different stages of development, of reproduction. So there's no really discrete stage. It's something that goes from a regular cell with a nucleus that dissolves its nucleus. All of a sudden you see chromosomes, and then they start to align, they move away, they do all these things until you make two cells. Okay, so you started with one cell and you ended up here with two cells. And those are the steps that we're going to look at in more detail. Now, why this is so very important is because really, if we knew exactly what triggered a cell to continue to divide and a cell to stop dividing, we would actually cure cancer. So cancer is making two cells out of one and making two more and two more and two more until you have a growth. You have a tumor, you have a cancer. So very important that we actually understand this. The stages that I really do want you to learn are the PMAT stages. And you have other videos from YouTube that I have listed for you. PMAT is kind of the crucial part of this cell division, right? Where you are seeing the chromosomes in a phase called prophase. In a phase called metaphase, these chromosomes are all aligned in the middle of the cell. A really weird thing. And they align beautifully. And then they're going to split down the middle. So if I could make a line here, I would be splitting them down the middle where one part is going to move in one direction and the other part is going to move away from itself in the other direction. Until I have a full pair here, full set of chromosomes in one cell, full chromosomes set in the other cell, and this is going to divide down the middle. Okay, so those are meta for being in the middle, ana Ana means away, so the chromosomes are pulling away from each other, and telo means end or finish. So that's the end. I want you to know, even though in the book you'll see early metaphase, later metaphase, I just want you to know this part. Well, if you looked at it individually, you'd have prophase. Oh, look, we have a nucleus, and we have these chromosomes floating around in the nucleus. P, from PMAT. Meta, here they are aligning in the middle of the cell. Meta means middle. Metaphase. Anaphase, they're pulling away towards the poles. So now I'm going to go back one. They were linked here, and we're going to have to slice them down the middle. And now they are sliced down the middle, and one set of chromosomes goes to one side of the cell, another set of chromosomes goes to the other side, ana meaning away. And finally, telophase, where you're forming two separate cells and you're starting to see the formation of the nucleus again. PMAT. Cytokinesis is when you finally have two separate cells, and now you don't really see the chromosomes so well anymore because they're not condensed. If they're not condensed and scrunched up together, you don't see them as these discrete shapes. Instead, you just see this sort of craziness here inside the cell. So those stages then are prophase, metaphase, anaphase, and telophase. Now before that comes another phase called the interphase, where the DNA is replicating before it actually starts its division. Okay, they'll condense in prophase, but at the early part of prophase. And then in late prophase, the nucleus is gone, chromosomes align, chromosomes pull away, and then we have two cells. This mouse of mine is too sensitive. What if the body loses control of this cell division? If we have too much cell division, where you have one cell dividing into two, and those two divide into two, and then divide into two, that's when we have our cancers. And that is that cell division out of control. So it's mitosis out of control is cancer. In this slide, I want you to look at the difference between a benign tumor and a malignant tumor. Notice that in the benign tumor, you have this capsule. So if I encapsulate those cells that divided too much, I have a little bump. And that little bump is really not creating any kind of problem because it is sort of encapsulated and out of any kind of pathology. It's not going to hurt you at all. However, a malignant tumor is when those cells keep reproducing, it's not encapsulated, and they can fall into a blood vessel or a lymph vessel that's part of your immune system and travel. And they will travel. That is called metastasis. Metastasis is when those cells fall into some circulatory system of your body, travel to a different part of the body, in this case, this bone marrow here, establish a new tumor. And this is what malignant tumors are. They have the ability to metastasize and move from one part of the body to another and create more tumors. And that is what is so very dangerous. Have we figured out how to tell these cells to stop reproducing? No, we haven't. And that is the clue to cancer. I'm going to stop there. The next video will be on meiosis.